Hi, good afternoon, everyone. My name is Sheena McCormick, and I'm going to present to you today FAPP2 promotes tumor cell growth in human colon cancer through activation of WNT signaling. These are my authors. So I chose this paper because I used to work for a gastroenterologist. We saw many patients that had colon cancer, and I've actually had some family members who was diagnosed with it. So I thought it would be nice to know if there's anything that can assist in preventing, other than the actual prevention of screening colonoscopies, what can be done. This paper talks about how FAPP2 cells are then created as knockout cells through the use of CRISPR-Cas9 to see if the knockout cells can help with the proliferation of colon cancer. And so what is colon cancer? Colon cancer is malignant cells that are in the colon tissue, and they begin as polyps. So as we'll see here in the colon, these circles here indicate the actual colon polyps that would be seen during a colonoscopy. And symptoms that you may have are a change in bowel habits, blood in the stool, diarrhea, constipation, gas, bloating, narrowing of stools, weight loss, fatigue, vomiting, several different symptoms that you may feel like just is involved with something else. So in colon cancer, you have 4 different stages. You have stage 0, 1, 2, 3, and 4. And the difference between these stages is where they're presented and how far through the layers have they gone. So you have your mucosal layer, which stage 0 has not increased through. Stage 1 goes through the mucosa into the submucosa. Stage 2 goes from the mucosa, submucosa into the serosa. And then stage 4, it goes through all the different layers, and that's when it actually starts metastasizing to the other organs and other areas of the body. So how do we diagnose and treat colon cancer? Colon cancer is diagnosed through rectal exams, fecal occult blood exams, barium enemas, colonoscopies. During a colonoscopy, if polyps are found, biopsies are then taken and ran through a pathologist lab, and they are then determined whether or not they are cancerous. So for a colonoscopy, a physician will insert a probe into the patient's rectum. It'll travel through the colon here. At the end of the probe, there is a camera and a light. The camera will allow the physician to see on the screen in front of them where they are in the colon, what they're visualizing, and if there is a polyp, then the physician would then insert a device that would remove the polyp, and they could send that off for pathology. So other treatments other than the removal of the polyps would be surgery if a colon resection is needed, radiofrequency, cryosurgery, chemotherapy, and ablation and radiation. So in this particular paper, as we mentioned before, it talks about FAPP2. FAPP2 stands for human phosphatidylinositol 4-phosphate adapter protein 2. It can be found on chromosome 7, and it is a 519 amino acid that encodes for the PLEC-HA8 gene, and that can be found on the long arm of chromosome 7 right here. Along with this, there is a C-terminal domain, GLTPH, that enables FAPP2 to transfer glycoceramide between the different compartments in the cell. And then there's the N-terminal pH domain, which stands for the plextrin homology domain. And this generates the FAPP2 to target the Golgi apparatus. So now that we know what FAPP2 is, what is the function behind it, and what is the purpose of it in this paper? FAPP2 is this protein right here. You'll notice some beta sheets and some alpha helices that are behind there. FAPP2 is a cytoplasmic lipid transfer protein that moves the glycoceramide from the Golgi apparatus to the ER. The goal of using it in this paper is to explain the expression and function of FAPP2 in colon cancer, since there's no other studies that have been found that explains the reason behind it. So to answer some of the questions that they had, they used different methodologies. I'm just going to cover just some brief ones that they used in this section, which was immunohistochemistry. You have your sample slide that contains your sample. Usually the sample has been previously embedded in paraffin wax to preserve it. The paraffin wax is then removed. You rehydrate that sample. You retrieve the antigen of interest. You will treat that with a primary antibody. Against the primary antibody, a secondary antibody is used against the primary. Then using a substrate, you'll cleave off your product, and then you're able to visualize your product using microscopy. In this study, 90 samples were analyzed that contained colon cancer tissue and adjacent non-tumorous tissue. Here you'll see two different samples. The colon cancer tissue is indicated with a C, and the non-cancerous is indicated with an N. And so the non-cancerous tumor tissue is basically the surrounding tissue of that tumor cell that did not contain cancer. So using the immunohistochemistry, they were able to use anti-FAPP2 antibody to detect the FAPP2 expression, which you see the staining right here, and all the cancerous cells, where the non-cancerous cells, there was no staining. And then that antibody was incubated overnight at 4 degrees Celsius, and then using a secondary antibody against that, it was incubated for another hour at room temperature, and then DAB substrate kit was then used to visualize the staining. Using that information, we were able to then put the information into a pie graph, and what it showed was that the expression of FAPP2 is upregulated in colon cancer, and 52 out of 90 samples were upregulated, and that was 57.8%. because they had stronger staining. 20 out of 90 had no difference. The staining appeared to be the same. And then 18 out of 90, which was about 20%, indicated that there was downregulation and that there was lower staining in the non-cancerous tissues. So then using CRISPR-Cas9, which is a gene editing tool, they were able to answer the question of what is the function of FAPP2 in colon cancer. So with CRISPR-Cas9, you have a guide RNA that associates with the DNA of interest. And then with this guide RNA, Cas9 enzyme comes in and associates with that. It cleaves the DNA interest of site, so then you get this. And your repair mechanism comes in and finishes the job to repair the mismatched DNAs. Then using Western blot, they also helped answer the question of what is the function of FAPP2 in colon cancer. So Western blot is the separation of proteins using SDS page, and this denatures the protein. You then transfer the proteins to a membrane. You incubate that with a primary antibody, and against the primary antibody, you'll incubate it with a secondary antibody, and then you analyze your results. And what they found was that using cancer cell lines SW620 and RKO, FAPP2 is involved in the regulation of colon cancer cell growth. Western blot confirmed this right here, as you'll notice in just the RKO and SW620 cell lines prior to the knockout cells, there were bands present indicating FAPP2. But when guide RNA was used for knockout cell lines, the bands diminished, indicating that FAPP2 is involved and that the gene editing was successful. So then they used a CCK8 assay kit to determine whether or not FAPP2 inhibits cell proliferation. So this assay kit uses a CCK8 reagent that is added to the cells. Those are then incubated using a measure optical density microplate reader at 450 nanometers. They then were able to detect it. And what that showed was that cell proliferation is decreased in the knockout of FAPP2. And this was indicated by after incubation of 6 days, your normal cell line, the knockout cells did show a decrease. And then as you move on to the RKO cell line, the knockout cells were decreased from there as well. In conclusion, FAPP2 decreases cell growth in colon cancer, indicating that FAPP2 upregulates colon cancer. Future directions: state that further studies need to be done with FAPP2's influence and the WNT beta-catenin signaling pathway, and these are my references. Thank